The bathyscaphe Trieste touched the bottom of Challenger Deep on January 23, 1960. Jacques Piccard and Don Walsh had spent 4 hours and 47 minutes descending through absolute darkness. The pressure outside their thick steel walls climbing past 1,000 times what it is at sea level. They had 20 minutes on the bottom. Through a small plexiglass viewport lit by quartz arc lights, Piccard saw something move. He described it as some type of flatfish resembling a sole about 1 foot long and 6 inches across, with 2 round eyes on top of its head, swimming slowly away through the bottom ooze. Walsh, in later interviews, called it a white flat, like a halibut or a sole, a foot long. Whatever it was, it crossed in front of the only window into the deepest place on Earth that any human being had yet reached, roughly 10,911 meters below the surface, about 7 miles down. And then the sediment cloud swallowed everything. Walsh described the view as like sitting in a bowl of milk. No photograph was taken. The 20 minutes ended. Trieste released its ballast and began the 3-hour, 15-minute climb back. What Piccard and Walsh reported seeing was, at the time, scientifically startling. Not because no one had believed life existed in the deep ocean. That idea had been abandoned since the 1870s, when HMS Challenger dredged animals from 3,000 fathoms and found them alive. What was in dispute in 1960 was something narrower: whether a vertebrate fish, with its bones, its proteins, its cell membranes, could function at hadal depth at all. The biology argued against it. At 10,911 meters, hydrostatic pressure exceeds 1,086 bars. The effect on unadapted cell membranes isn't the dramatic crushing implosion that popular imagination supplies. It's quieter and more total. Pressure forces phospholipid bilayers, the structural fabric of every cell wall, to compact into a gel-like phase, losing the fluid mobility they need to function. Ion channels stop working. Membrane proteins seize. The cell doesn't burst, it simply stops. Simultaneously, high pressure drives water molecules into the hydrophobic cores of proteins, eliminating the internal cavities that maintain their 3-dimensional shape. Enzymes unfold. Quaternary protein structures begin to dissociate at around 100 to 200 megapascals. The molecular machinery of an unadapted organism shuts down long before it reaches hadal depth. There is also the matter of calcium carbonate. The Pacific Ocean's carbonate compensation depth, the horizon at which seawater becomes so undersaturated with carbonate ions that calcium structures dissolve faster than they form, lies at roughly 4,000 to 4,500 meters. Challenger Deep sits more than 6 kilometers below that line. Any carbonate shell, any calcified bone reaching that depth dissolves. The trench floor receives no skeletal remains from the surface. What falls as marine snow, the constant drift of dead plankton, shed mucus, fecal pellets, fragmentary organic material, arrives as soft particles, not hard structure. The water itself sits at approximately 1 to 4 degrees Celsius throughout the hadal depth range. Cold alone does not stop life, but combined with extreme pressure, it suppresses enzymatic reaction rates and metabolic processes in ways that compound the challenge for any organism whose biochemistry wasn't specifically adapted for that environment. The consensus before 1960 was that teleost fish, bony vertebrates, simply couldn't survive at these depths. The biochemistry didn't permit it. So when Piccard described what appeared to be a flatfish moving across the floor of Challenger Deep, it seemed to challenge that consensus directly. Except that it probably didn't. Not in the way the description implies. Marine biologist Torben Wolf, writing in the early 1960s, found the sighting somewhat dubious. He pointed out that flatfishes are predominantly found in shallow and mid-depth water, that no flatfish had ever been collected from anything approaching 11 kilometers, and that flattened sea cucumbers, holothurians of the species Galathiothuria aspera, white, oval, roughly a foot long, had been confirmed at great depths and could easily be mistaken through a small, sediment-clouded viewport in artificial light by two men who were, as Walsh later acknowledged, not ichthyologists. In 2012, marine biologist Alan Jameson and biochemist Paul Yancey published a systematic analysis in the Biological Bulletin on the validity of the Trieste flatfish, dispelling the myth, listing 5 lines of evidence against the identification. The mismatch with known flatfish depth ranges, the severely restricted visibility window, the total absence of fish in all subsequent Challenger Deep surveys, and a biochemical argument that makes the flatfish identification not merely unlikely, but physiologically impossible. That last argument rests on something specific. As depth increases, fish must accumulate higher and higher concentrations of trimethylamine N-oxide, TMAO, a molecular stabilizer that counteracts pressure-induced denaturation of proteins. TMAO increases with depth in teleost fishes, approximately 261 millimoles per kilogram at 4,800 meters in abyssal grenadiers, rising to 386 millimoles per kilogram in Hadal snailfish collected from 7,000 meters in the Kermadec Trench. Yancey and colleagues, writing in the Proceedings of the National Academy of Sciences in 2014, extrapolated this trend to its logical end. At roughly 8,200 to 8,400 meters, a fish's internal osmolality would reach the same level as seawater itself. At that point, the osmotic gradients driving all cellular fluid regulation flatten to zero. No fish, their analysis concluded, should be able to function below approximately 8,200 meters. Challenger Deep sits 2,700 meters below that limit. What Piccard almost certainly saw was a flattened holothurian, a sea cucumber, not a vertebrate, not a fish, moving through the ooze. It remains the closest thing to direct visual evidence of complex life at that depth from the 1960 dive. There is no photograph. There is no specimen. There is only a 20-minute observation through a clouded viewport made by 2 men who described what they thought they saw and later admitted they could have been wrong. A sighting isn't a survey. An observation isn't an inventory. What the trench does contain, confirmed by decades of subsequent research, is extraordinary. It's simply not what the flatfish story promised. A 2017 paper in the journal Zootaxa by Geringer, Linley, Jameson, Goetze and Drazen formally described Pseudoliparis swirii, the Mariana Hadal snailfish, based on 37 individual specimens collected between 6,898 and 7,966 meters in the Mariana Trench. This small, translucent, gelatinous-looking fish isn't what most people picture when they imagine the world's deepest vertebrate. It survives in part through TMAO. Muscle concentrations in specimens near 8,000 meters reach almost 400 millimoles per kilogram. Its genome, analyzed in 2019 in Nature, Ecology and Evolution, shows adaptive changes in pressure-tolerant cartilage formation and modifications to phospholipid bilayer composition that keep cell membranes fluid under pressures that would gel and immobilize the membranes of any surface fish. The holotype specimen, catalogue number USNM 438975, was collected on November 21, 2014 at 7,949 meters. In 2017, a Jamstek and NHK team filmed a Pseudoliparis snailfish at 8,178 meters, a film record, not a captured specimen. The current deepest fish record is 8,336 meters, set in 2022 in the Izu-Ogasawara Trench by an unnamed species from the same genus. No bony fish has been confirmed beyond approximately 8,400 meters, which aligns closely with what TMAO accumulation predicts as the biochemical ceiling. Above that horizon, the trench is occupied. The amphipod, Hirondelia gigas, a crustacean, has been captured at Challenger Deep itself, between 10,897 and 10,900 meters, surviving partly by synthesizing an aluminium shield from gluconic acid derived from ingested wood fragments, a chemical solution to the dissolving carbonate environment it inhabits. Giant single-celled xenophyrophores, some reaching several centimeters across, have been documented at depths up to 10,641 meters in Serena Deep through 2011 dropcam studies and confirmed at Challenger Deep by video footage from James Cameron's 2012 expedition, though their species-level identification awaits specimen collection, which hasn't yet been achieved. Microbial cell densities in Challenger Deep surface sediments run to approximately 9.7 million cells per cubic centimeter. A 2025 analysis of 1,194 trench samples identified 7,564 microbial species in the Mariana system, roughly 89% of them previously unreported to science. Life is there. It has adapted across geological time to every chemical challenge the environment presents. It uses TMAO to stabilize its proteins against pressure. It redesigns its cell membranes for conditions that would kill a surface organism within minutes. It relies on marine snow, that perpetual, slow fall of organic material from the sunlit ocean thousands of meters above, as its primary food source, concentrated by the V-shaped trench geometry into a narrowed funnel of detritus. The trench is biologically active. The question the research forces is different. How much of what is actually there have we ever directly seen? Before April 2019, 3 individual human beings had descended to Challenger Deep: Walsh and Piccard in 1960, James Cameron alone on March 26, 2012, reaching approximately 10,908 meters and spending around 3 hours on the bottom. As Jameson noted in the Marine Technology Society Journal in 2020, following the 1960 Trieste dive to the Challenger Deep, it was 52 years before Deep-Sea Challenger followed suit. Between those 2 events, there were no crewed visits, not one. The most biologically singular place on Earth was visited twice in half a century, for a combined bottom time of less than 4 hours. The 5 Deeps expedition between April and May 2019 changed the pace dramatically. Victor Vescovo and his team completed 4 dives to Challenger Deep in 8 days using DSV limiting factor, setting a new depth record of 10,928 meters on the first dive on April 28th. By early 2024, around 27 individuals in total had descended to Challenger Deep. Sources differ, and some analyses suggest the number may be marginally higher. Even accepting 27, the total accumulated human bottom time at the deepest point on the planet amounts to a few dozen hours across more than 6 decades. But the access problem goes deeper than visitor count. Each deployment of a baited camera lander, the principal instrument for deep hadal surveys, frames a field of view of approximately 68 by 51 centimeters. That is roughly 0.35 square meters of seafloor visible to the camera at any given moment. Challenger Deep alone comprises 3 basins, each several kilometers long and approximately 2 kilometers wide. A single lander deployment surveys less than a postage stamp fraction of a single basin's floor. Alan Jameson, who pioneered full ocean-depth hadal lander systems and has deployed them over 800 times in the world's deepest trenches, has produced an unmatched dataset. And still, the coverage, measured against the total trench area, is vanishingly small. The instruments also see only part of what lives there. Baited camera systems are structurally biased. They attract scavengers. Animals that come to carrion appear on film. Animals that don't, non-motile epifauna, suspension feeders, cephalopods, non-scavenging predators, are invisible to the method. Jameson's own published work on cephalopod depth records states directly that establishing depth limits for squid and octopus is hampered because contemporary hadal sampling has favored baited traps and baited camera systems, to which cephalopods, being predatory, are rarely attracted. The instruments illuminate one feeding gill with clarity. They pass silently over the rest. Deep-sea trawling from surface vessels adds its own distortions. Nets sample across depth as they ascend, mixing fauna from different levels into a single catch. And the recovery problem compounds everything. Hadal-adapted cell membranes are engineered specifically for pressures that don't exist at the surface. When a specimen ascends, the pressure drop causes those membranes, stable at depth, unstable at surface pressure, to invert into non-lamellar structures. Many animals disintegrate before reaching the ship's deck. What arrives, if it arrives at all, may not resemble what was alive below. Metagenomic approaches have been applied at Challenger Deep with genuine results. 586 metagenome-assembled genomes from 26 bacterial and archaeal phyla in a 2022 Nature Communications analysis. But these methods, as applied so far, have focused on microbes and viruses. A comprehensive environmental DNA survey capable of detecting fish, invertebrates, and echinoderms, the kind of broad macrobial inventory attempted in some shallower trenches using 18 srRNA metabarcoding, hasn't yet been conducted comprehensively at Challenger Deep. The trench is still hiding things then, in a specific and defensible sense. Not because anything is being concealed, not because a promised creature lurks undiscovered in the dark, but because the tools deployed have been narrow-field cameras with a structural bias toward one feeding guild, brief, crude visits measured in hours, and specimen recovery methods that frequently destroy what they recover. The gap between what is being confirmed and what might exist isn't a gap filled with speculation. It's a gap created by the geometry of observation itself. Someone will inevitably mention megalodon at this point. Otodus megalodon was a warm, shallow-water coastal predator whose last fossil evidence dates to approximately 3.6 million years ago, feeding on large marine mammals in tropical shelf environments where neither the temperature nor the prey exist at hadal depth. The trench isn't hiding a megalodon. There is a more substantive comparison worth considering. 12 human beings walked on the surface of the moon, all of them between 1969 and 1972, during the Apollo program. For 59 years after the Trieste's 1960 dive, that number exceeded the total of 3 people who had ever visited Challenger Deep. The equation has since shifted. By early 2024, roughly 27 individuals had descended to the deepest ocean, a number that likely surpasses the lunar surface count. Whether that represents a misallocation of resources or simply reflects the different returns of each frontier is a genuine disagreement. Ocean exploration has produced chemosynthesis, novel pharmaceutical leads, detailed climate records from deep sediment cores, and a microbial biodiversity whose dimensions were almost entirely unknown until recently. Space exploration produces data on planetary formation, cosmic radiation, and conditions for life elsewhere that can't be gathered from a submersible viewport. The two programs draw from largely separate funding streams and scientific communities. Presenting them as a simple trade-off flattens a genuinely complex question. But the parallel does illuminate something real. The moon has been systematically mapped. Its surface has been photographed at resolutions that allow individual boulders to be catalogued. Challenger Deep, a feature roughly 48 kilometers long at its full extent, has been directly imaged at close range for a total of perhaps a few dozen hours across all dives in history, with cameras that frame less than half a square meter of floor per deployment. That is the actual mystery. Not a creature concealed in the dark. Not a secret the ocean is keeping. The plain, structural fact that the deepest place on Earth has been approached through apertures so narrow, for durations so brief, with instruments so specifically biased, that comprehensive knowledge of what lives there remains beyond what current methods can produce. The confirmed life is real and remarkable. A translucent fish, chemically engineered to exist at pressures that would dismantle a surface organism at the molecular level. Single-celled organisms the size of a small coin, confirmed only by video because no specimen has yet been recovered. Microbes catalogued for the first time in 2025, the vast majority of them unknown to science before that analysis. Those facts don't require embellishment. The honest unknown, the life that may exist beyond the edge of a 0.35 square meter camera field, the species that don't come to bait, the organisms that dissolve before they reach the surface, is a more compelling mystery than any invented one. Which unanswered question about the deep ocean, in your view, most deserves the next expedition's time and resources?